Hello everyone, Mr. Linder here. Let's get started with the nervous system. As you go through the next set of videos, you're going to be working on one of the most complex systems that's found in the human body, especially from a physiological standpoint. So as you go through the nervous system, it's always a good idea to review the anatomy before you get into all of the major physiological mechanisms that take place within the nervous system. Now, I don't have time to go through all of the anatomy with you, but I will show you a few slides just to get you guys thinking about the anatomy of the nervous system. So as you read through textbooks and as you watch videos on the nervous system, you're always going to come across various vocabulary. Everything from action potentials to glial cells to rough endoplasmic reticulum, sensory neurons, and so forth. But there's also synonymous or synonyms that go along with various terminology that's used. So when you hear things like nerve impulse, you still want to think action potential. When you hear something like cytoplasm of an axon, we're also talking about the axoplasm. When you hear soma of a neuron, you're actually talking about the cell body of the neuron, and so forth. So it's always a good idea to go back and review a lot of your anatomy of the nervous system just so you don't get lost with the vocabulary. As you study the nervous system, you know that there's a hierarchy to the nervous system. The nervous system is composed of the central nervous system. So we have the brain and the spinal cord, but then it's also composed of the peripheral nervous system, where you have sensory neurons that travel through nerves and are going to connect to the central nervous system. You have motor neurons that are going to leave the central nervous system, and they're going to carry information out to either somatic controlled structures or autonomic controlled structures. The somatic controlled structures are going to be your skeletal musculature, and then your autonomic is going to be subdivided into sympathetic and parasympathetic, and they're going to control cardiac muscle, glands, smooth muscle, etc. So take a look at the hierarchy of the nervous system, again, just so you don't get lost in the vocabulary and the structure and the makeup of the system as you try to learn the physiology. There are various types of neurons within the nervous system. The neurons carry the action potentials or the electrical signaling. There's sensory type neurons, there's interneurons, there's motor neurons. We also use names like bipolar neurons, multipolar neurons, and then of course we go through the structure of neurons as well. So this is a diagram just showing you some of the different varieties of the neurons, from the multipolar varieties to the bipolar varieties, unipolar ones. So take a look at the various structure so you have a general idea of the differences in neurons within the central and peripheral nervous system. And then basic structures of the neuron, so dendrites of the neuron, the cell body or soma, the nucleus of the neuron, the axon hillock, the axon terminals. Notice here there's Schwann cells wrapping around this particular neuron, creating what's called the myelin sheath. So lots of anatomical things to take a look at before you dive into the physiology. And again, various diagrams to take a look at. This is actually looking at transport through a neuron. So production of, let's say, neurotransmitters using the Nissl bodies or the rough ER that's found in the soma, transporting vesicles over to a Golgi, and then we have axonal transport along the axon down to the axon terminal. So we can generate neurotransmitters in the soma. We can transport them down to the axon terminal, and then we can use exocytosis to release those neurotransmitters. And those neurotransmitters can then be used to communicate with the next neuron in a pathway. So take a look at some of that axonal transport information. Glial cells, these are the supporting cells of the nervous system, so they support the neurons, and they have a variety of roles as well. So ependymal cells being involved in the movement of cerebral spinal fluid, astrocytes, blood-brain barrier, recycling of neurotransmitters, all kinds of different functions, microglial cells, phagocytic cells, oligodendrocytes. These are myelinating cells in the central nervous system. And then in the peripheral, we have the Schwann cells and the satellite cells, which play a supportive role. So again, take a look at all of the neuroglial cells that are found within the nervous system as well. So here's a nice diagram that shows you a neuron, and then we have associated glial cells, ependymal cells, microglial cells, astrocytes, oligodendrocytes, and so forth. In previous videos, I went over the Nervest Equation. So take a look at resting membrane potential and the Nervest Equation. And so just as a review, the Nervest Equation allows us to calculate the equilibrium potential for a particular ion. And so I showed you guys how to do equilibrium for potassium or how to calculate equilibrium for sodium. And this helped us to establish where resting membrane potentials would fall for human body cells. If equilibrium for potassium is at negative 90 millivolts and equilibrium for sodium is at positive 60 millivolts, then resting membrane potentials for cells are going to fall somewhere in between there, since these are the main ions for resting membrane potential. And it turns out that most cells fall into the negative 85 to negative 60 millivolt range for resting membrane potential. As we now move into the nervous system, we're going to see how we can change the resting membrane potential and actually do work. Because it turns out that nerves and neurons and muscle cells are excitable tissues. And so they can respond to stimuli, they can propagate electrical signals, and then they can send those electrical signals constantly and over long distances throughout the body. And so the Nervest equation is always a good equation to review when you're thinking about the nervous system. The other one to review is the Goldman-Hodgkin-Katz equation. That one actually allows us to calculate what the resting membrane potential would be by looking at multiple ions. So instead of doing hypothetical equilibrium for one ion, we can use the Goldman-Hodgkin-Katz equation to look at permeability and then also look at multiple ions at a time. So here we have potassium, sodium, chlorine, and then we can come up with the voltage of the membrane, or essentially the resting membrane potential. So it's a much more complex equation, but it's more powerful because it allows you to actually calculate resting membrane potentials. But normally, we don't use numerical values when we're trying to talk about what's happening with the resting membrane potential. We don't usually say, the resting membrane potential moves from negative 70 millivolts to positive 30 millivolts. Instead, we use vocabulary to describe what's taking place. So it's more of a conceptual thinking. We'll use terms like depolarization or hyperpolarization or repolarization. So if we're not using essentially numerical values, even though you can and we could graphically look at these things, if we're going to use vocabulary, what does the vocabulary actually mean? So when we talk about the term depolarizing event, we're talking about an event that's happening with the cell where it's taking the membrane potential in a more positive direction. So on this graph, if this is resting membrane potential for a particular neuron, negative 70 millivolts, if there's a depolarizing event taking place, then the membrane potential is going in a more positive direction. So we can see on this graph that we're moving from negative 70 up towards, let's say, negative 40 millivolts. If we're going in a positive direction, then it's called a depolarizing event. Commonly, we see sodium involved in depolarizing events. So if we have a cell and we allow sodium to enter into that cell, because sodium has a positive charge associated with it, and the inside of the cell is inherently negative, outside of the cell is positive. If we begin to add positive charges through channels into a cell, that's going to take us from a negative value towards a more positive value. And so that's known as a depolarizing event. We also have hyperpolarizing events. If this is negative 70 millivolts at resting membrane potential, and we see the value of the membrane voltage going in a negative direction, then we call that a hyperpolarizing event. Hyperpolarizing events are oftentimes caused by the movement of potassium, but we also see chlorine involved quite a bit in hyperpolarizing events. So the way that would work, if you have a cell, and again, the inside is negative, if potassium leaves the cell, so if we open up channels that allow potassium to leave the cell, we are losing positive charge from the cell, and so that's going to make the cell more negative. And so if we become more negative, it's a hyperpolarizing event. How does chlorine come into play? Well, chlorine has a negative charge, so if chlorine, which is in high concentration outside of the cell, enters into the cell, so we're going to add negative charges to the inside of the cell, that's going to make the inside of the cell more negative. So again, that's a hyperpolarizing event. The other term that you're going to run into is repolarization. So after you've had a depolarizing event, we'll notice that the membrane potential is returning to normal, and so that event is known as a repolarization. And repolarization is caused by the movement of potassium ions. So we typically see sodium associated with depolarizations, potassium associated with repolarizations, and then we typically see potassium and chlorine associated with hyperpolarizations. Now, that doesn't mean that there can be exceptions to these, because there will be. We'll see calcium get involved in some events when we talk about muscle activity, but this is the norm that we see with most of the cells, especially neurons, when we're talking about how we depolarize, repolarize, and hyperpolarize. Now, it's important to note that when you change the membrane potential, it doesn't mean you're changing the concentration gradients. So just because a particular ion, sodium is high concentration outside, low concentration inside, just because we allow some sodium to come into the cell, creating a depolarization, that doesn't mean that the concentrations have suddenly reversed. We're still going to have a high concentration of sodium outside and a low concentration of sodium inside. We're not reversing the concentrations just because we're seeing a change in membrane potential. In fact, very few ions have to move in order to cause large changes in membrane potential. Because if you recall, we're actually measuring the membrane potential in millivolts, okay, thousandths of a volt. So it only takes 1, 2, 3 ions to start making a change in millivolt values. The next thing I want to take a look at is channels that are involved in the nervous system. So how does a cell change its ion permeability? Well, simply put, we can open and close existing channels within the membrane of a neuron in order to change the permeability for that particular cell. So when we look at the neuron, in particular, we can be looking at the dendrites or the cell body, okay? Or we can be looking at the axon. So again, we want to look at and know our structures. Okay? If we're looking at, say, the dendrite or the cell body, or even the axon, in order to change the permeability, we're going to talk about changing the channels that are found within the plasma membrane. And so by opening or closing channels, we can affect the permeability for a neuron. So if this is a neuron, all we need to do is change the permeability of a potassium channel or of a sodium channel or a chloride channel or a calcium channel. And then we can do that to change the overall permeability of the cell. So the main types of channels that you're going to see in the neuron are sodium, potassium, calcium, and chlorine. Now, some channels will let multiple ions go through. And so you'll see situations where you might have a channel that opens where both sodium comes in and potassium goes out. So depending upon which electrochemical gradient is the greatest, that'll determine what type of change is going to take place. Are we going to get a depolarization or are we going to get a hyperpolarization? That's going to depend on the electrochemical driving forces for those particular ions. Another term we use frequently when we talk about ion channels and movement is conductance. And conductance measures the ease at which ions can move through a channel. Conductance is oftentimes influenced by whether, of course, the channel is open or closed, but also whether the channel has a gate or not. So in this particular diagram, we can see that this channel right here is just a hole, and so ions can flow freely through it. But there are some gated channels where it might be open at some point in time, but then there's this ball and chain structure that can close the opening, and therefore ions cannot enter through the plasma membrane. So gating a particular channel will give us some control in what's taking place along the plasma membrane of a neuron. So we look at conductance not only with the ease at which they move, but also how we control the gates as well. Most gated channels are going to fall into one of three categories. You either have mechanically gated ion channels, so they open in response to physical forces like pressure and stretch. We see this a lot with sensory neurons. We have chemically gated ion channels. These are going to open in response to neurotransmitters or neurohormones. And this is going to be involved in a lot of the electrical signaling that we see taking place, especially synaptic transmission. So the release of a neurotransmitter from an axon terminal, release of a neurotransmitter, and then binding to a receptor, let's say on a dendrite. And so we have a receptor, and so it receives that neurotransmitter. And so we have this chemical gated channel right here. This receptor is a chemical gated channel, and it's dependent upon the neurotransmitter that binds to it. And then we have voltage gated channels, and voltage gated channels open in response to changes in the plasma membrane. So if you can change the resting membrane potential along the membrane, you can affect voltage gated channels. For example, if you have an axon and you have a particular channel, let's say we're dealing with a sodium voltage gated channel. The sodium voltage gated channel is closed at negative 70 millivolts. But if there's a signal that changes the resting membrane potential, let's say from negative 70, let's say it changes to, let's say negative 40 millivolts. Turns out that at negative 40 millivolts, sodium voltage gated channels are open. And so if this channel opens because of a change in voltage, sodium then can enter through that channel. So reaching certain voltages can affect whether the channel is opened or closed. If it's not a gated channel, then oftentimes we use the term leak channel to describe the channel and how the ions are moving through it. But it turns out that not all leak channels are actually leak channels. And what do I mean by that? If you have a cell, oftentimes we just have channels which are just holes in the membrane. And so sodium can leak through, or potassium can leak through. But it turns out not all leak channels are actually leak channels. Oftentimes leak channels are a form of voltage-gated channel. They just happen to be open at resting membrane potential. So there are different kinds of channels that we see along the membrane of a neuron. So we have to get used to classifying what they are. We have leak channels, we have voltage-gated channels, you're going to have chemically regulated channels, and so forth. So get used to all the different types of channels that are associated with the nervous system. The voltage-gated channel that I was just talking about, the sodium voltage-gated channel, is affected by what we call the threshold voltage. And so threshold is a key term you need to know. Threshold is the value that has to be reached in order to open a certain type of voltage-gated channel. Sodium voltage-gated channels have a threshold of negative 55 millivolts or more positive. So what do I mean by that? Negative 55 millivolts or more positive. It means that the resting membrane potential has to change from negative 70 to at least negative 55 or more positive, negative 54, 53, 52, negative 50, negative 40, and so forth. It has to be negative 55 or more positive in order for that sodium voltage-gated channel to open. If you don't get to that number, it'll stay closed. So if this is a sodium voltage-gated channel, at resting membrane potential, negative 70 millivolts, we see that the channel is closed. In order for it to open, you have to reach threshold. You have to reach threshold. Negative 55 millivolts or more positive. So if there's an electrical signal that's moving along a dendrite, these are found, sodium voltage-gated channels are found along the axon of a neuron. So we'll put a sodium voltage-gated channel here. Sodium voltage-gated channels are found along the axon. So let's say we have an electrical signal that's moving along a dendrite and along a cell body, and it's on its way towards the axon. And let's say when it gets to the axon hillock, you're only at negative 60 millivolts. The change was from negative 70 millivolts, okay, to negative 60 millivolts. If you're at negative 60 millivolts, you're not going to open the sodium voltage-gated channel because you haven't reached threshold. You haven't reached that negative 55 or more positive. But if you have a dendrite, cell body, and an axon, remember resting membrane potential, negative 70 millivolts, and let's say the signal when you reach the axon hillock is negative 50, then you have reached threshold. You are more positive than negative 55, so you can open those sodium voltage-gated channels, and you can see an influx of sodium into the axon. And that's actually what's going to trigger what we call the action potential, and we'll get into that in future videos. So it's really important to understand these voltage-gated channels and how they function, because that's going to dictate whether you can get action potentials or not. Sodium channels, turns out, are faster than the potassium channels. So sodium channels are going to open at a faster rate than potassium channels, but they actually close faster as well. So potassium channels take a little bit longer to open, but then they stay open for a longer period of time. To finish off this sodium voltage gated channel, you can see this ball and chain structure. And this ball and chain structure is what's going to lead to the inactivation of this sodium voltage gated channel. So if the ball and chain is fitting into the opening of the channel, it's disrupting the channel. So even if sodium was to try to enter into the cell, it cannot. It's going to hit the ball and chain structure and then it's going to bounce off. So inactivation will prevent a neuron from responding for a certain amount of time until that ball and chain structure moves, and then we get back to the original closed state that we see in resting membrane potential. So that is our sodium voltage gated channel. So inactivation is going to stop the movement of ions through a particular channel. Regardless of what the ion is trying to do, it can't penetrate through the membrane until that ball and chain structure is out of the way. As you continue to look at the nervous system, you need to understand Ohm's Law. And Ohm's Law looks at current as it relates to voltage and resistance. So I is the current. So in terms of electricity, we look at current. V is voltage, and R represents resistance. And just from a basic standpoint, if you can understand current and voltage and resistance, then you can really understand how the nervous system is operating, because the nervous system has to obey the properties of Ohm's Law. So what does that mean? It means that you're going to get better current when you increase the voltage. There's a direct proportionality. So as you increase voltage, you will increase current. If you decrease voltage, you're going to decrease the current. There's a direct proportion, and that's true within the nervous system. If you increase the resistance in the nervous system, so let's say there's more resistance in the cytoplasm of the neuron, you're going to decrease the current. That's an inverse proportionality. Sometimes it's easier to tell if you write the equation like this, I equals V over R. Okay? So you can see I and V, we can put a 1 here, are on the top, and I and R are on opposite sides of the fraction. And one's on the numerator, one's on the denominator. So if you increase resistance, you're going to decrease current. But if you have less resistance through the cytoplasm of the cell, then you will have better current. And so it turns out that neurons are going to follow these electrical values as they obey Ohm's law. So I equals V over R is an important concept to learn. So as we change the voltage, or as we change the resistance of a particular cell, that will affect the current of the action potential as well as the graded potential as it moves through the neuron. So what is an action potential and what is a graded potential? So first off, let's talk a little bit more about resistance, and then we'll define graded potentials and action potentials. So resistance is really looking at the cytoplasm. So when we look at resistance, we focus a lot on the cytoplasm. Turns out that the lipid bilayer is normally a good insulator. And so the plasma membrane does not typically allow things to move across it. And so it typically has high resistance across the membrane. And then if we look at the cytoplasm, let's say the axon is narrow versus an axon that is wide, you're going to have more resistance as you compact all the components of the cytoplasm in a small cell, and you'll have less resistance when you can spread things out in a larger cell. So the bilayer itself, the phospholipid bilayer, creates a level of insulation and resistance, and then the cytoplasm creates a level of resistance. So how do we get past that? How do we get better conductance through the nervous system when you deal with these things? Well, that's where the channels come into play. To get across the bilayer, we have to have the voltage-gated channels, whether they're sodium voltage-gated channels or potassium voltage-gated channels. So opening, closing channels is going to improve our conductance. And then the size of the cell itself. Okay, the diameter of the cell matters in terms of reducing resistance so that information can travel. The other things that we'll look at too is myelination of the nervous system and how that can improve the current flow through the nervous system. Let's get back now to the idea of different types of electrical signals. So there are graded potentials and there are action potentials. Action potentials I'll cover in another video. But to finish up this one, let's talk about graded potentials for just a second. Graded potentials are the depolarizing or hyperpolarizing signals that occur typically along dendrites and cell bodies as they then move towards the axon hillock. So graded potentials are typically along the dendrites and cell bodies. The membrane potential is going to change based on the result of opening certain types of chemical gated channels or mechanically gated channels. And so those are going to trigger the graded type potentials. So if we look at this table, we have graded potentials versus action potentials. And again, action potentials will be in another video. Focus more on that. But graded potentials, if we look at where they typically occur, they occur along the dendrites and the cell bodies. Now, why is that? Well, a graded potential is really a signal that oftentimes is based off of, let's say we have a receptor in the dendrite, and oftentimes it's a neurotransmitter that binds to that receptor that's going to open this chemically gated channel, and let's say it lets some sodium in. Well, if it lets sodium in, it's going to cause a depolarization. That depolarization is not an action potential yet. It's just what we call a graded potential. But that graded potential is changing the resting membrane potential. What once was negative 70 millivolts is now going to be a different value. Maybe it changes the dendrite to minus 30 millivolts because enough sodium has come in to create that more positive value in the membrane potential. So graded potentials are going to happen typically along dendrites and cell bodies. They typically happen when you open chemically gated channels or mechanically gated channels. Sometimes there'll be voltage gated channels, but oftentimes it's chemical or mechanical ones. What's interesting though is graded potentials get weaker with distance. And that's a big key. Weaker with distance. That's in essence why they're called graded potentials. Okay? They get weaker with distance. They don't stay the same. Okay? They don't have the same energy, if you will, the whole time. Think of it as kind of a downhill. Okay? They start out strong. Graded potentials start out strong, but they get weaker as they travel through the dendrite and as they travel through the cell body. So they get weaker with distance. Action potentials are not going to get weaker with distance. Action potentials are going to maintain their amplitude. They're going to maintain their strength. And we'll talk about why in future videos. So what creates the graded potential then? Well, it's ions entering or leaving the neuron. So sodium coming into this dendrite causes a depolarization. Let's say we have a dendrite cell body, we have a receptor, and let's say it's a neurotransmitter that binds the receptor, and chlorine comes in. Well, since chlorine has a negative charge, that's a hyperpolarization. Depolarizations have a chance to create action potentials. Hyperpolarizations are taking us away from threshold, away from that negative 55 millivolts, so they're not going to lead to an action potential. They're going to shut off the signaling pathway. Large stimuli cause larger graded potentials. And then, of course, graded potentials get weaker with distance. So how does a graded potential spread then? If you take a look at this diagram, you'll see we have an axon terminal, and this is where you can release, let's say we release a neurotransmitter that's binding to this receptor, we'll call this R right here, and it's triggering this chemically gated channel to open, and it lets the sodium come in. So the sodium is going to enter at this initial location right here. But what it's going to do is it's going to spread throughout the dendrites and throughout the cell body, because it's going to move from positive areas, because the sodium that comes in is positive, to areas of the cell that are still more negative. Because remember, the rest of the cell is at negative 70 millivolts. And maybe this area is currently at minus 30 millivolts because positive ions have come in. So these positively charged sodiums are going to start to move by local current flow and be attracted to areas of negative charge. But as the sodium is attracted to other areas of the dendrite and cell body, it's going to get weaker with distance, because not all of the ions are going to go to the same location. If I was to draw in a few of my own ions here, more sodium, okay? Some of the sodium is going to come over here. Some of the sodium is going to go over here. In fact, some of the sodium might be pumped back out by a sodium-potassium pump. And so not all the sodium is going to make it to a particular area, and so it's not going to cause as great of a change. So although the change at the initial location was minus 30, we might only be at minus 50 millivolts at this location over here. And we might only be at minus 55 millivolts at this location down here, because we're getting weaker as we move away from that initial influx of sodium. So graded potentials get weaker with distance. The more channels you open, the stronger the change is going to be, and so the graded potential can start out stronger, but it will still get weaker with distance. So again, why do they get weaker with distance? Well, you're going to lose ions to the outside. They're going to leak out of the intracellular fluid back out into the extracellular fluid. And then, of course, the cytoplasmic resistance and the movement of the diffusion of the ions through the cytoplasm is going to be difficult depending upon how many things it has to get through. And so you're not going to get as many ions spreading throughout all of the cytoplasm. And so you're going to get weaker with distance. But here's the big key take home then. Graded potentials are either going to get us to threshold or not get us to threshold. So in this example, we have an example of a graded potential starting at the cell body on this particular neuron. So some neurotransmitter has bound to a receptor. Let's say it's letting in sodium again here, causing a depolarization. We're actually graphing it so we can see the depolarization. So initially, it's a strong depolarization. It gets to close to negative 40 millivolts. But as the sodium diffuses through the cell body, the signal is getting weaker. We can see it as we measure the signal at this location right here. We see that the depolarization is at negative 55 millivolts. which is good. That's the threshold value if you were at the axon hillock, but you're not at the axon hillock yet. The axon hillock is down here, the trigger zone. So we're at negative 55 in the middle of the cell body, but we're not at negative 55 at the axon hillock, because as you continue to move towards the axon hillock, and we measure at this location, we notice that we're only at maybe negative 60 millivolts. The graded potential has gotten weaker with distance. And so what's happened is we have gotten to a point where we're below threshold or sub-threshold. If you're sub-threshold, you can't open the sodium voltage-gated channels. So sodium voltage-gated channels do not open. And if they don't open, then you're not going to trigger an action potential. So no action potential. So even a graded potential that causes a depolarization is not a guarantee for an action potential. In this case, there was no action potential. We know for sure if the graded potential hyperpolarizes, takes us even farther away from threshold than we started, then we're definitely not going to get an action potential. So how do we get an action potential? Well, we need a graded potential that's going to be supra threshold. So we need a supra threshold graded potential. So here we have release of neurotransmitter. We bind to a receptor with the neurotransmitter. Let's say, again, we let in sodium, and we're letting in a lot of sodium. And so we got a much bigger change in resting membrane potential. It took us from negative 70, original resting membrane potential, to negative 30 millivolts. And now as the graded potential gets weaker with distance as it spreads out, okay, as we measure it in the middle of the cell body, we see that we're still above threshold. We're at minus 40 millivolts. And then when we get to the axon hillock, the trigger zone, we're still above threshold. There's that negative 55 or more positive. Right, we're going in a positive direction. So we're at minus 50 here. And that's enough to open sodium voltage-gated channels. And if you can open sodium voltage-gated channels, you can let sodium into the axon. So this is a sodium voltage-gated channel. Oops. You can let sodium come into the axon, and now you can trigger an action potential. And so we'll pick up in future videos on what is an action potential and how do you transmit the action potential along the axon. So I hope you enjoyed the introduction to the nervous system. Take care.